We are continuing with foundational concept number 1 of biology, section 1A, proteins and amino acids, structure and function. Just a reminder that this section is worth 55% of the biology section. This video is about amino acid descriptions and the classification of hydrophobic or hydrophilic amino acids. Just as a precursor, all of the information provided in this video is important to remember for the MCAT. The AAMC often provides pKa values except for basic and acidic amino acids. These videos include testable facts and memorization aids. It's important to memorize structures and basic traits of amino acids along with the shorthands of amino acid names. And as an important reminder, you will be tested on amino acids, so be sure to study them often. So first, we'll start with hydrophilic amino acids. Hydrophilic means water, hydro, and loving, philic. These amino acids often create hydrogen bonds which can only be formed with fluoride, oxygen, and nitrogen when they are attracted to hydrogen. These elements are so electronegative that they withdraw electron density from the hydrogen. So hydrophilic and charged amino acids are addressed in the following video: acidic and basic amino acids, and they include aspartate, glutamate, histidine, lysine, and arginine. Hydrophilic amino acids are polar, meaning that there are differences in the electronegativity between the bonded atoms. So this delta sign up on the right indicates that something is partial. So in the image of H2O, there is a partial charge on the hydrogen and a partial charge on the oxygen. As you can see, because the oxygen has much more electronegativity and also is a larger atom, this partial positive charge is pulled towards the oxygen, and it creates kind of a V or a bent shape. You'll learn more about this in the chemistry section. For amino acids, in order to identify a polar group, it is easy to look for OH, alcohol, NH2, amino, or SH, thiol groups in these side chains. So the hydrophilic and polar or neutral amino acids include asparagine, glutamine, serine, threonine, and cysteine. So asparagine is abbreviated ASN and N. So necessary information is that hydrogen bonds at the NH2 group. This is the animated form of aspartic acid, so that means that the amino group replaces the acid's OH. So some tips to help memorize this structure is that asparagine has one N in the R group, just like there's one N in the name. Asparagine is an ammonia, so NH3 derivative, which makes urine stinky, just like asparagus. For glutamine, it is abbreviated GLN or Q. So one way to help memorize this is pronouncing glutamine as glucutamine. It's important for muscles, immunity, and getting QTs is another way that I remembered it, because it tells you all of the important functions. Some necessary info for the MCAT is that hydrogen bonding occurs at the NH2, and it is the animated form of glutamic acid. So again, the NH2 replaces the acid's OH group. Serine is abbreviated SER and S. Necessary info includes that it hydrogen bonds and it is phosphorylated at the OH. It participates in the biosynthesis of purines and pyrimidines for DNA and RNA, which are going to be talked about in later videos. To help memorize this, I think of serine like SO, just like in SOHCAHTOA, or second. I think two groups are included. When you see OH on an amino acid group, think of phosphorylation. So the next few amino acids are all going to be phosphorylated ones, and as are the acidic amino acids, glutamic acid and aspartic acid. So threonine is abbreviated THR or T. It also hydrogen bonds and can be phosphorylated at the OH. It's composed of three groups, CH, OH, and CH3, like serine, but it has an additional CH3. When you see, and like we said before, this can be phosphorylated, and just remember that phosphorylation occurs when there's an OH group in the R chain. The last hydrophilic amino acid is cysteine, which is CYS or C. It creates sulfur bridges in proteins via disulfide bonds at the SH. So what happens is two SH groups line up with one another, and the hydrogens are lost and the sulfurs bond onto each other. This amino acid is the only one found in the D configuration. And to help memorize this, cysteine has two groups, so CH2 and SH. It's pronounced cysteine as in cysteine. The S stands for sulfur. And cysteine stabilizes proteins with sulfur bridges. Hydrophobic amino acids are nonpolar, but they are broken down into two different groups that are more specific. So yeah, hydrophobic and nonpolar ones include valine, leucine, isoleucine, methionine, and proline. And the hydrophobic and aromatic amino acids are phenylalanine, tryptophan, and tyrosine. Nonpolar means there is a minimal difference or a balanced electronegativity. So nonpolar solutions, such as oil, cannot be dissolved in polar solutions like water. So as you can see from the image, there's two carbon atoms that are double bonded to one another, and they create a straight line, which also indicates that a bond is nonpolar. Right, so first we'll talk about valine, which is abbreviated VAL or V. So like I said before, it is nonpolar and hydrophobic. It is a very small amino acid. Its carbon chain is in the shape of a V, as you can see on the image, and it's all just made out of carbon. So it is known as a branched chain amino acid along with leucine and isoleucine. Again, leucine is abbreviated LEU and L. It is also nonpolar and hydrophobic. And you think of it as a Y shape because it's valine plus an extra CH2 group. Like mentioned before, it is a branched chain amino acid. Isoleucine is an isomer of leucine, so it has the same groups as leucine, but it just has swapped position at the branch. And it is the final branched chain amino acid. Methionine is abbreviated MET and M, and it's very important to remember that this amino acid indicates the beginning of a protein during translation with a start codon AUG. This is the only 3 start codons that you should know for the MCAT. It is one of 2 amino acids with sulfur, the other one being cysteine. So the way that I think about it for memorization is methionine is mandatory for life, because without methionine we would not have any proteins being synthesized. That is, its pure purpose is to indicate the beginning of a protein. Met indicates methyl, so a CH3 group, and thi indicates sulfur. So that also helps with memorizing the last bits of this amino acid. The way that I remember the start codon for the MCAT was thinking of it as augment, because it is used to change the mRNA to protein. Proline is an amino acid often tested on the MCAT because it has a unique shape where the amino acid side chain actually turns back to meet the amino group found in the amino acid backbone. So because of this shape, it can create kinks in a protein chain, and it is important for recognition and signaling within the body. It is also found within the alpha helix of DNA. So the easiest way to memorize the structure is that when you look at the image, it actually looks like a P for proline. So now we're going to talk about aromatic amino acids, which are also hydrophobic because they have a large carbon ring, and carbon is a hydrophobic atom. Phenylalanine is abbreviated PHE or F. So I think of this as pronouncing it with an F sound, so phenylalanine. The chain is a phenyl group, so which is a 6-membered benzene ring minus a a hydrogen plus an alanine from the very beginning of this video. So some necessary information is that it is aromatic, it is large or bulky amino acid, and that it's not processed well by those with PKU, which causes neurological symptoms, as this amino acid will build up in their brain. Tryptophan is the largest amino acid. It is very hydrophobic because it is so large, and this is all carbon, which is hydrophobic. It does not hydrogen bond well because of its large size. To memorize that, it is represented with a W and also its shape. Think of pronouncing it with a W sound, so tryptophan, as the side chain does look like a W if you cut off the top lines. The next amino acid is tyrosine, which is TYR or Y. So it is the least hydrophobic aromatic amino acid, as the OH group can be phosphorylated. So because of this, sometimes people will consider it a hydrophilic amino acid, but the large carbon chain makes it more hydrophobic. To help memorize it, I think of it, the group, as being large like a tire, spelled Tyre, or that it is phenylalanine with an OH group attached to it. So lastly, we're just going to touch on the two small amino acids, which can fit really well into amino acid chains without kinking them. They're essentially neither hydrophobic nor hydrophilic because they are so small, although some people will consider them hydrophobic because they lack polar elements and have carbon or hydrogen in their side chains. So glycine is the smallest amino acid, and it only has an H as R group, meaning that it is the only non-chiral amino acid, and that's because there's two hydrogens attached to the alpha carbon, which is the carbon in green. So the easiest way to memorize this is that G comes before H alphabetically, which is all that's in the chain. Alanine is the second smallest amino acid, and it can be wedged into tight loops or chains. One easy way to remember the structure is that A is the beginning of the alphabet, and a C group is the beginning of all other amino acids except for glycine.